In the interest of time, I'm not going to talk to you today about the loss of soil caused by farming. We see from 10 to an 18 times rate of our soil loss. I'm not going to talk to you about what's happening to the rest of the soil, desertification, soil salinization, pollution through chemical runoff, or new subdivisions and industry. Not going to talk to you about we're also losing water in our aquifers. That only 60% of the world's aquifers are being depleted faster than they're being filled. Not going to talk about the energy change that we have in growing food. It used to be in the 1940s, it only take 2.3, one fossil fuel calorie to make 2.3 calories of food. Now, we only get 1/10 of a calorie of food for every calorie of fossil fuel. That's a 23 times reduction. We see, not going to talk about nitrogen fertilization increasing over this from 1960 by three times, while agricultural land has stayed pretty much the same. Or phosphorus increasing, doubling. Of what happens to that nitrogen and phosphorus, we only get 10 to 30% utilization. The rest of it goes into our atmosphere, into our waters, into the oceans. We see that the reactive nitrogen on this planet has increased. You can see all the farming areas on this planet. Also, what happens with this nitrogen? With our health, upper respiratory disease, asthma, cancer, birth defects, cardiovascular disease, sudden infant death syndrome. Phosphorus is being applied on this planet at 10 times the assimilation that this planet can take it in. You can see it also in the many areas. What does all this give us? Well, Lake Erie last year, or 2 years ago, the LSS in China, the Baltic Sea, the most polluted body of water on the planet, and of course Lake Okeechobee in Florida. And then its release into the Gulf of Mexico and the red algae blooms to pretty much destroyed the whole tourist season this year in Florida. The documented dead zones on this planet. This should cause us pause of what we're doing. Same thing with global pharmaceutical consumption, chemical industry outputs, and global pesticide applications. 6 times increase since the 1960s. What's being correlated with that is increase in multiple sclerosis, Crohn's disease, type 1 diabetes, and asthma. Autism has increased 84 times since 1975, from 1 in 5,000 to 1 in 59 on our children. What's closely associated with that is our use of glyphosate. Farmers in the U.S., farm cash income is the same as it was in 1960. Farm debt has increased 14 times. Farm leverage is the same it was back in 1980. Our return on invested cash, we've had a 3 times decrease. This is not a viable business model. So the challenges we have ahead: produce higher quality and more nutrient-dense food on declining land area, using less water, fewer energy and natural resources, do all of this under difficult economic circumstances, with soils and ecosystems that are continually being degraded. We have a challenge in the future. We're not going to change this by fighting the existing reality. This thing is too big and too set, and the money flow is too much towards that system. The only way we're going to change this is to bring a new model that makes that system obsolete. We need to understand that living organisms have shaped every facet of this planet. They have accomplished this by doing 2 things: improving the efficiency of the capture, storage, and utilization of energy that we capture from the sun, and from increasing system biodiversity. Let's look and see what's happened over the past, since this planet has been here for about 4.6 billion years. Water hit here about 4.2 billion years ago. Bacteria came relatively soon after, about 4 billion years. cyanobacteria and anoxygenic photosynthesis. One of the greatest movements towards the changes that we see on this planet is the ability to capture that energy from the sun and store it and utilize it later. Nitrogen fixation popped up about 3.2 billion years ago. Oxygenic photosynthesis, another great adaptation on this planet, started to give us an oxygen-rich atmosphere. Fungi, after our atmosphere had oxygen, eukaryotes came in. Fungi, about 2.2 billion years ago. The ozone layer and multicellular life, 1.6. Mitochondria, the ability to increase the efficiency of energy conversion, 16 times. First single-cell animals, about 750 million years ago. Land plants, about 700. Glomeromycota, the fungi that associate with plants and help them harvest nutrients and water from the soil, came in at 400 to 500 million years ago, and we're here. We're the new kids on the block. We benefited from all that's come before us. The last 600 million years, you can see right now, we're here about 400 parts per million on our atmospheric CO2. You can see it's been higher. It's been up to 7,000 parts per million on this planet. You can see we're at about 14 degrees C. You can see also global temperatures have gone above 25 degrees C. You can also see oxygen on this planet has varied. We're sitting at about just a little less than 20% at this point. Along the bottom, you see all the different extinction events and their influence on the different gases, CO2, atmosphere, oxygen, and temperature. How life has changed this planet. Our first land plants, as I said, about 700 million years ago. Cambrian explosion, that's when we had corals and shelled animals start to pull the carbon, the calcium carbonate out of the seawater to make their shells for protection. You can see the dramatic decrease in atmospheric CO2. Glomerulaceae, again, came about right about here, another drop. Lignin, vascular plants, evolved right in this area. Coral reefs have come and gone in the fossil record, and they had an influence on reducing atmospheric CO2. 8-meter-tall fungi, progenitosperms, roots, leaves, and seeds. This allowed plants to colonize the planet, to move away from the water bodies. And you can see that was our Carboniferous period. Significant amounts of energy were stored that we're using right now. Coal seam development, peak O2. One of the reasons that we were able to store that carbon up is nature did not have a lignin-degrading microbe. Well, along came a microbe that could degrade lignin. You can see an increase in atmospheric CO2 here. Gymnosperms also evolved in here. Conifers, ginkgos, evergreens, see another drop. Angiosperms, the flowering plants, you see another drop of atmospheric CO2. Coral reefs, you'll see here we had a cooling period. Landmass positioning changes. Mammals evolved. Ungulates, your grazers, also evolved. And grasslands. We did not have grasslands till 55 million years ago. And it's the action of the ungulates, the grazers, and the grasslands that have built up, or had built up, the carbon in the soils and gave us the soils that we have today. We need to understand how critical the elements are on this planet. Now, also understand the influence of biology on the accessibility of these nutrients. Iron. In the Earth's crust, it is not in sufficient population in order for us to mine it economically. It's where the microbes in the sea attracted iron to their cell walls. They built up enough mass that they would sink into bands. We mine those bands of iron. It's all resulting from microbes. Aluminum has to go through 2 microbial processes before it can be bauxite. That's the economically viable way we can mine aluminum. Copper. We're now finding out that over 60% of the copper that we mine has actually been deposited by microbes in anaerobic water seams. Of course, carbon. We all depend on that. It's our energy resource. We use it today. We're the benefactors of all that's come before us. all of these, everything we have around us, the concrete we have here would not be available without the calcium carbonate, or the limestones that built up from from microbial life. The copper, all our electricity is transported on, aluminum as well. Everything around us, our roads, the asphalt, it's all microbially oriented. We need to understand how important microbes are in our life. This is the Earth's crust element of abundance. You can see by the size of the circle, its abundance. Nature took advantage of that. You can see all the ones that are in great abundance, you see life has used them. But like Finian said this morning, we're about 96.2% gas. My wife declares often I'm more than that. But you can see every element that's in pretty good quantities, life evolved to use that. You know, there's one exception here, selenium. It's very low in the Earth's crust. It's only about 50 parts per billion. In the human body, we require it at 200 parts per billion. If we don't have that, we lose our immune function. We are susceptible to viral infections. Our reproduction is not as great. Mood, thyroid function, cardiovascular disease, and cancer, just from a depletion of selenium in your body. We're now seeing the potential that there are 3 mass extinctions that we cannot explain. In a recent study looking at the selenium amount in the sediments of the oceans has concluded that once ocean selenium concentrations drop below 1 part per million, it lines up exactly with extinction events on this planet. One element is all it takes. Elements that we use in our body, like in our heme, where it allows us, the hemoglobin, to hold on to oxygen or CO2. A simple change in pH of this iron molecule in this protein structure allows us to have 70 times the carrying capacity of oxygen in our blood, allowing us to be the size we are. Otherwise, we'd be like microbes. But you can see we have 100 trillion of those in our body, made every second. There's 280 million of those in each red blood cell, and we have 20 to 30 trillion red blood cells in our body. Nature used the same structure for chlorophyll. You can see just a change from iron to magnesium. Nature's pretty unique. Hydrological cycles. Do they depend on microbes? There's one that happens to float around in the atmosphere, Pseudomonas syringae. First of all, I need to tell you that water freezes at minus 37 degrees C, pure water. Everybody's been taught 0 degrees, that's where water freezes. It doesn't freeze without something to nucleate on. This particular bacteria has in its cell wall structure a protein that allows supernucleation of ice crystals on its body. You'll find it in almost every snowflake. You'll find it in almost every raindrop. So it's very important. Simple biology is important in the hydrological cycle of this planet. We have a large number here. It's not the number of questions that a 10-year-old asks its mom every day, although that was the consensus. It's not our national debt. Yours, I think, is right in here. It's not the number of seconds since life first evolved on this planet. It's not the number of sand particles in all the beaches on this planet or the number of stars in this known universe. It's the number of biochemical reactions that goes on every second in your body. So if anybody tells you that you're not doing anything, tell them you're busy. We need to understand that microbes are the backbone of every organism and ecosystem on this planet. You have about 10 trillion human cells. You have about 90 trillion microbial cells. You're outnumbered almost 10 to 1. You have 1 quadrillion resident viruses. Do not underestimate that viruses are part of this. This is a system that we evolved in. You have about 20,000 human genes. Your microbial complement has 2 to 20 million microbial genes. What do? They digest your food, do nutrient generation, cell renewal. We used to think they were 60% of our immune system. We're now figuring they're about 80%, maybe more. They control your appetites and cravings. They synthesize vitamins that you can't get in your diet, detoxify carcinogens, prevent allergies and skin diseases, turn on and off genes that regulate brain development, anxiety, depression, autism, arthritis, and emotional behavior. What happens when these microbial communities are disrupted through antibiotics or diet? Almost every autoimmune disease that we have today is related to the dysbiosis of our microbiota. Again, we were talking earlier about multiple sclerosis, Crohn's disease, type 1 diabetes. Crohn's disease and IBS and C. diff infections have increased dramatically. What they're finding, though, that if you get a healthy inoculum from another individual and do a fecal matter transplant, and this is great before lunch, sorry, that people that have suffered for decades with this, that have had operations, resections of intestines, that have tried every avenue, and some people have died from this, that some of them within 24 hours, they can be cured with the right biological community restored back into their system. So we're kind of saying, oh, let's look at the gut microbiota diversity of your hunter-gatherer and agriculture versus industrial. Here's hunter-gatherer, here's the beginning agriculture, here's our industrial diet. We're very different in our gut microbiota, and that's a lot of the reason we have the problems we have. Looking at some diet-induced differences in gut microbiota, Tanzania, Malawi, and Venezuela, much more diversity in the system. And diversity is key. Diversity is the currency of survival on this planet. Us in the USA, very low diversity. See what happens when we take fiber out of our diet? Generation after generation after generation after generation, we lose the diversity in that system. Okay, just put the fiber back in the diet. The top one here relates to what happens when we put the fiber back. Nothing. We get extinction events in our stomach microbiota from what we eat. You do a fecal matter transplant, it's immediately black and functioning. So what we're thinking is, this works in humans. We know what we've done in agriculture has destroyed the microbial community. Can we do this in agricultural soils? Can we just do an inoculation of microbes to bring these systems back? Let's look at how nature has this set up. There's a ladder of succession that is developed on this planet, from a bare soil parent material to your old growth forest. You can see there's a change from a predominantly bacterial to predominantly fungal, as you also increase net primary productivity from about 90 grams of dry biomass up to 2,200 grams. This is what a bacterial dominant soil looks like. Layer after layer of bacteria. That's a California rice field. This is what a healthy agricultural soil should look like. You have your organic matter here with fungi that have completely covered it. You have your bacteria. This is what the Muir redwood forest in California looks like. You can see a lot of organic matter, very few bacteria, and that's covered with fungi. We, in agriculture, are here. We do weeds really well. Now, weeds are a misnomer. Weeds are there for a purpose. They are there to improve that soil, to move that soil up to the next level. But we keep disturbing it, and we keep having weeds. We need to be here, about 1-to-1, in order to grow the crops that we like to eat. But we also have to understand, to stay at that point on the ladder, agriculture is controlled disturbance. This is what nature used to develop those deep carbon, high carbon percent soils. The bison were kept in tight herds by the predators. They were moved along pretty quickly. They could only harvest about 30% of the forage that was available. They would stomp the rest of it in. They would deposit their dung. The dung would be rolled up into small balls by dung beetles, placed in the ground, basically composting in place. So what you have is this herd of inoculators going across the prairies. inoculating every time they go past. Does this work if we start to bring this system back? This is Nancy Rainey, Corona, New Mexico, about 12 inches of rain a year. In the last decade, she's gone from 3 species of grasses to 45. She's reduced her medicine bill by 90%, reduced supplemental feed bills by 66%. We had originally built swales to capture water for stock tanks. She changed the dynamics of the soils so much as far as infiltration and holding capacity that they no longer fill up. Her aquifer water tables are rising, and her grasslands recovered in a total drought. Alejandro Carrillo, in the Chihuahuan Desert, south of us, about 9 inches of rain. He's improved his plant diversity, his stocking rate, bird diversity, and native animal population. He's changed. This is where I live. Beautiful, huh? Green, lush. This used to be, when the Spaniards came through, belly high to a horse with grass. And yet, with our grazing techniques, we've completely destroyed it. But what he's done is bring it back. He went from 250 acres per cow, to 20 acres to support 1 cow, and restored the grasslands. This, in order to implement this into agriculture where we did not have the animals, we developed a composting process. Most of the composting is done with windrow, and you do not end up with a very fungal-dominant compost. This is a static composting system. It's no turn. It's the lazy man's way to do it. It's available on YouTube if you want to do this. We can talk about it tomorrow on one of the classes that we'll have. But it solved one of the big problems with dairy manure for USDA. It dropped the salinity of that substrate from 33 to 40 millisiemens down to less than 3. But most importantly, it gave a fungal dominant and biologically diverse compost. This is the texture of that compost. It's a lot like a clay. Looking at the change in the biology through the composting process, you see an ebb and flow of different phyla of microbes. Some start strong, go to nothing. Some are practically nothing and end up to be a dominant player at the end. These are 2 composts made a decade apart with completely different materials using the same composting process. You can see striking similarity between the end product. We see at 4 weeks this kind of diversity in the top 80% of the microbial community. At 22 weeks, when everybody says it's done, let's put it out. Okay, this is 60 weeks. There's a 4 times increase in diversity over this time period if you allow this compost to mature. It becomes an inoculant at that point and not a nutrient amendment. Well, we had to look at some growth tests, looking at 4 or 8 different composts that were available in our area. And these 2 right here are composts that we grew our chili plants in. You can see there's significantly more plant productivity in the compost we had. I was originally an NPK junkie. I'm about 14 years reformed now. Because I thought, okay, nitrogen, phosphorus, and potassium, it's all about that. Nitrogen didn't correlate to plant growth. Phosphorus did not correlate to plant growth. Potassium didn't correlate to plant growth. Okay, I was like, all right, it's got to be organic matter. No. Organic matter is important, but it is not what's going on here. We had looked at a fungal to bacterial ratio. Biology is key. Let's look at what happens as you move from a bacterial dominant, low carbon soil to a high carbon, fungal dominant soil. Where does the carbon that a plant captures go? You can see in a low fertility, low carbon, bacterial dominant soil, 3% of the energy that plant captured and turned to photosynthase goes to the plant. The rest of it went into the soil. 97% of the energy that plant captured was pushed into the soil. It must be important for a plant to do that. That's not a good survival technique. As you move up, you can see 17%, 21%, 30% into the root, shoot, and fruit of the plant. all the way up to 56%. Now, we're right here in agriculture. Low carbon, bacterial dominant, we're about 11% efficiency in this system. We should be able to, according to this, increase the productivity of these soils 5 times. Does it work in the real world? That's in the lab, that's scientists, okay? First of all, you need to understand that tropical rainforest, the most productive ecosystem, terrestrial ecosystem on the planet, does about 2,200 grams of dry biomass per square meter. Cultivated land does 600. We water it, we fertilize it, we weed it, and that's about the best we can do with what I'm calling a biologically enhanced agricultural management, bringing the microbes back into the system. We're matching that 2,200. In an advanced situation where you've built your soil carbon up, we're seeing up to 3,200 grams. So half again, more productive than the most productive ecosystem on the planet. Biologically enhanced agricultural management, BEAM. We started out by applying the compost we made at 800 pounds or 400 pounds per acre. That's 45 grams per square meter. That's what it would look like on a square foot. It's a sprinkling. There's hardly any nutrients here and very little carbon. So what did we do with net primary productivity with that? And we did this one time at the beginning. We didn't do any more applications after that. Just get it inoculated, support it with plant growth, always having a cover or a commodity crop on it. This is our control, and this is after 1 year beam. We went from 50 grams per square meter to 250 grams per square meter. That's already a 5 times in 1 year. That's not good production, though. That was just on a winter cover crop. This is year 7. We're at 1,141 grams. We've almost quintupled the second after that 1 year of beam. This is a summer cover crop that came after that. It was black oilseed sunflower, it was Sesbania, it was a forage soybean, and a few others. It's about 4 to 6 inches tall on June 23rd. A month later, you can see it's about 4 to 5 feet tall, and that's normal for black oilseed sunflower, about in here. This is 17 days later. It's 7 feet tall. Black oilseed sunflower heads like this, larger than my head. My wife says that's pretty big. This is Sesbania. It's a crop native to southeastern California, southwestern Arizona. It's a legume, and it also associates with mycorrhizal fungi. The first season we grew it was 4 feet tall. The second season was 6 feet tall. This is the third season, 12 feet tall. We're changing the dynamics in this soil as we pump the photosynthetates in to feed these microbes and have them start to develop. This is a sandy desert trial. On this trial, we grew cover crops continuously. We cut them, hauled them off, and replanted. Same inoculation, the 400 pounds per acre, first year. We wanted to see how much can we tax this system? How much can the biology do? Well, this is the second year. The first year was 800 grams of dry biomass per square meter. This is the second year at 1,608 grams. This is the third year at 2,190 grams. Remember, 2,200 grams, most productive ecosystem on the planet. This is a crop that you plant October-ish. It doesn't get but about this high. And then mid-February, it wakes up. And then for the next 10 weeks, we see this kind of growth. That's 10 weeks of growth matching the most productive ecosystem on the planet with biology. This is what it looked like as we started the first 4 seasons, from uninoculated to inoculated. This is what it looked like over the 3 years. We were doing about 3,200 grams compared to 2,200 grams without. Now, you don't have to inoculate your soils. You can bring them back with cover crops. But if you extrapolate these out, you're going to match it at year 30. So it's your choice. What we saw on this was 777 pounds of nitrogen per acre, or 872 kilograms of nitrogen per hectare per year fixed in the above ground biomass. Now, a lot of scientists have trouble with this, but with the right biology, this can happen. Also, we saw an increase of 5 times on this plot as well, from 500 grams up to 3,100 grams. This is the starting soil. Lovely, huh? Sand, rock, gravel. No A horizon. This is the control. After 3.5 years, we had a 3-inch A horizon. We had a 7-inch A horizon in the compost treated. I'm a molecular biologist. I like to look at all the species of microbes and what they do. This is an analysis of those 3, the soil we started with here, the desert, the control, and the compost treated. There was about 193 species of microbes that popped to the top. Either they were linearly correlated with the biomass, or they were 100 times the population of any other organism. Now, what we saw with these microbes was they had the ability to fix nitrogen, both free living and symbiotic. Nitrogen cycling and carbon cycling. It's critical for us to understand that this is a system that cycles. Nothing is static in it. We want to think of this carbon as static, but we cannot. It is part of the process. It's a part of the life process. It's the energy flow. We're the same way. We're no different. We have a carbon flow within our system. We see metal oxidation, microbes that can oxidize metals. There's one in here that can do gold. Now, if it can oxidize gold, the most inert element on this planet, what other element do you think it'll have trouble with? None. We see phosphorus solubilizing bacteria. In our systems, the amount of phosphorus we've added that gets tied up because of the biochemistry, or the physical chemistry of the situation, is 40 years of supply. And yet we keep adding phosphorus. We have phosphorus solubilizing bacteria that free that up and make it available to the plant. We have antibiotic and antimicrobial production. So these plants can also put out, using the microbes, antibiotics to fashion the population the way they want it. We see biofilm and quorum sensing, carbon monoxide oxidation, microbes that degrade pesticides and xenobiotics, and phytohormone producing microbes. These are some of the free-living and symbiotic fixing nitrogen-fixing microbes. These are the nitrogen cycling and N2O degradation, carbon cycling, metal oxidation, phosphorus solubilization, antimicrobial antibiotic production. These are the pesticide degraders. We need these. What can they degrade? About anything we've put out there. There's malathion, chlorpyrifos, polyaromatic hydrocarbons, pyrenes, toluene, naphthalene, vinyl chloride. That's plastic. So, aflatoxins, chlorinated ethynes, phytohormone production. There must be a significant interest that plants do well if a microbe is going to put out phytohormones. I kind of see plants as the first attempt at artificial intelligence by microbes. We're the second attempt. Not so good on that one. They didn't think it all out. In the US, we have food safety requirements. Escherichia coli just happens to be the marker microbe that we use. As you'll notice, as we improve these soils, the incidence of Escherichia coli goes away. We start to have inhibitive competition by microbes as we bring the whole system back. Quorum sensing and biofilm formation. I did mention that briefly. Quorum sensing is either when one microbe or a community of different microbes get together as a community. And they can express genes as a community that they can't as an individual. Kind of like us when we get in a quorum, we can do things as a community that we can't do as an individual. It's no different. We probably learned it from them. It can be used in symbiosis or virulence. That's how we get sick. You get a certain number of microbes that get together in a community and they can express genes that put toxins out. Competence, conjugation, antibiotic production, motility, and sporulation, biofilm formation. What we saw in these soils as we brought the biology back was an upregulation in quorum sensing genes of bacteria working together in a community. much over just your housekeeping genes, like ribosome formation. Ribosomes take the messenger RNA that's translated from your DNA, and they turn it into proteins. And you are proteins. You're either structural proteins or you're metabolic proteins. So housekeeping genes are extremely important. That expression level of about 1,800 hits for ribosomes compared to quorum sensing at 6 to 10 times. Thank you. Significant. As we bring these microbes back, as we get the community structure back, we'll start to see these systems be more functional and more productive. It gets very difficult in this world very quick. Protein interaction networks, you can pull on this string here, you could have 5 to 10 to 100 different reactions happen in some other place. Let's not forget the viruses. 800 million of them cascade onto this planet every square meter every day. They found out in the ocean, if you remove viruses, that food chain collapses. So this is a system that we're bringing back, and all of these are a part of it. What are the benefits observed in regenerative agriculture? Pulling this biology back into the system, we're seeing soil carbon increase. First 4.5 years of growing these cover crops, putting everything back in, looking at it from the perspective of, if I'm going to be a carbon farmer, I want to make as much on that carbon and get it in as fast as I can. Putting everything back, we saw 10.7 tons of carbon per hectare per year increase in our soils. That's about a 0.25% increase in carbon per year in the top 30 centimeters. Most scientists are seeing from 0.5, 0.2, or 0.7 from West, Nigley, and Law as far as our capability to increase soil carbon. But bringing the biology back, we get 1 order of magnitude more. We see the potential of doing about 19.2. That would be about 0.5%. Now, as a scientist, I was out on the limb saying something like this. But thanks to Gabe Brown, also you see at that rate, it would only take 50% of the arable land on this planet to capture all anthropogenic CO2. Gabe Brown came to the rescue. He struggled the first 13 years. He only got about 0.1% increase doing no-till, crop diversity, cover crops. Not till he brought in multi-species cover crops and livestock integration that he changed to over 0.5% increase per year. He did that when he hit 3% soil organic matter. That's one of the keys. And he stopped using nitrogen fertilizers. Now, Will Harris in Georgia, he has seen 0.36% increase with he implements his cattle into his farming operation. He's gone from 0.75 to 7.9%. Increased soil carbon use efficiency. They're saying respiration-wise, the more microbes you have, the more soil carbon you have, the more respiration you have. They're going to say it's either linear or exponential. It should be exponential. We had an 18 times increase in beginning soil carbon mass in this experiment. We had a 5 times increase in microbial biomass. That should have been exponential, but you can see that it levels off. We only had a 4 times increase in the soil carbon respiration mass. This is Will Harris's. He has a very similar curve with very similar results. 10 times increase in beginning soil carbon mass and only a 4 times increase in respiration mass. When you look at this a little different way, the percent of initial soil carbon respired in a bacterial-dominant, low-carbon soil was 55%. And yet, as you move up that chain up to a fungal-dominant, high-carbon soil, only 11% of that carbon is respired. This is how nature did it. This is how nature built up the carbon in the Great Plains. We talk about mineral nutrient depletion in our food systems. You can see what's happened since 1940. What we saw in the first 20 months of this BEAM study was a 1,100% increase in both manganese and iron. Magnesium, calcium, zinc, and copper availability increased 83 to 40%, and macronutrients also increased. Now, our soils are about 8.2. Both manganese and iron should have been decreasing in availability just from the physical chemistry. We see increased soil water storage capacity. See, the first 1% increase in soil carbon was a 5 times increase in water storage. And improved crop yields. We see a doubling over conventional production of cotton and chili. Organic, we love you, but you take a hit because you're trying to mimic conventional agriculture by hauling nutrients in. You need to get the biology in to furnish the nutrients for you. This is conventional cotton, 180 pounds. That's after the 1.5 years of beam. This was cotton in 2017 on that field after 7 years. 6-foot-high cotton. Doesn't mean you have cotton, unless you do. We did about 5 bales per acre, about 3,300 pounds of seed per acre. That's about a $2,000 an acre crop. Chili, 2018, about 17 to 21 tons. Like I showed in the previous one, we about doubled the productivity. This is in Dihaggerty in Australia, where they have taken this compost. I found out they're making compost the same way. They're using the same technique that we have. They adopted the Johnson-Su bioreactor. They apply it 1 kilogram per hectare with an extract that they shoot into the furrow when they plant the seed. They're getting 3 tons of barley in a region that normally gets 1.5 to 1.6. Now, this is the most telling slide on my presentation. This is the harvested barley. What came up exactly where they inoculated was a C4 grass they hadn't seen for 3 decades. Yeah. So once we bring the biology back, we don't know what these systems can do. So we can meet the challenges. We can rebuild the degraded soils and increase soil fertility. We can produce more nutrient-dense food and forage. We can use less water, because if we're doubling crop productivity, we can farm on half the land and get the same amount of crop. We can reduce energy use by allowing nature to extract and fix the nutrients we need. We can reduce both agrochemical pollution and atmospheric CO2 at the same time, and could improve productivity and profitability. Why should we pursue this? There is no planet B.